Hello, welcome to the biologist. So let's start. So today we are going to discuss about Mendel's pea plants, experiments, and about the process. And let's see how far we can go today. Okay. So why peas? That is Pisum sativum, which is the scientific name of the peas, because it can be grown in a small area, produce lots of offsprings, produce pure plants when it's allowed to self-pollinate several generations, and it can be artificially grown. How amazing is that? Reproduction in flowering plants. Pollen contains sperms produced by the stem. Ovary contains eggs found inside the flower. Pollen carries sperm to the eggs for fertilization, which is called the self-fertilization. Self-fertilization can occur in the same flower. Cross-fertilization can occur between flowers. Mendel's experimental methods. Mendel hand-pollinated flowers using a paintbrush. He could snip the stamens to prevent self-pollination. He traced traits through the several generations. How Mendel began. Mendel produced several strains by allowing the plants to self pollinate for several generations. So, first, what he did? First, he took the male anther and put it into the ovary. Okay, put it into the female stigma. Okay, so from which the germination occurred. So he took eight pea plant traits. What are those traits? Seed shape, which can be round or wrinkled, seed color, yellow or green, pod shape, smooth or wrinkled, pod color, green or yellow, seed coat color, gray or white, flower position, axial or terminal, plant height, tall or short, flower, purple or white. So, one thing you have noticed that the first character is written in capital letter because the first character represents the dominant character, and the second characters are written in small letters because the second characters represent the recessive character. Okay, so these are the dominant characters and these are the recessive characters. Did the observed ratio match the theoretical ratio? The theoretical or expected ratio of plants producing round or wrinkled seeds is 3 is to 1. Mendel observed ratio was 2.96 is to 1. The discrepancy is due to statistical error. The larger the sample, the more nearly the results approximate to the theoretical ratio. Generation gap. Parental P1 generation, the parental generation in a breeding experiment. F1 generation, the F1 generation or the first generation offspring in a breeding experiment, the first filial generation from breeding individuals from the P1 generation. The F2 generation, the second generation offspring in a breeding experiment. Second filial generation from breeding individuals from the F1 generation. So, if Mendel crossed a tall plant with a small plant, he will get a hybrid, which is uh, in the genotype, that is a capital T and a small t. But the phenotype will be all tall because the dominant characters always show their characteristic, not the recessive ones. So we took two pure plants, that is, what is pure? That is homozygous trait. That is, both the characters will be the same. That is, both will be capital T, another plant will be small t. And he got capital T and small t. And if he again crossed the if he crossed the hybrids, then he will get three tall plants and one short plant. That is, if he self-crossed the heterozygous plants, then you will get three tall plants and one short plant. That is, two will be of homozygous characters. That is, uh, these two will be of homozygous character and one be of heterozygous characters. So always remember, in any of the crosses, two, that is, one will be of a dominant homozygous and another will be of recessive homozygous. So in all the crosses, one cross will be dominant homozygous, another cross will be recessive homozygous, which will be pure. Okay, it is for all the crosses. So let's learn about monohybrid crosses. P1 monohybrid cross. Trait: seed shape alleles, which is capital R, round, small r, wrinkle. Crosses: round seeds and wrinkle seeds. So when we cross round seeds and wrinkle seeds, we get capital R smaller, capital R smaller, capital R smaller. So all the genotypes we get is uh, hybrids, that is capital R and smaller. When we cross pure lines, that is capital R and smaller smaller. So the ratio will be all alike and the genotype will be all the same, that is all the seeds will be round. Okay, this is monohybrid cross. The P1, homozygous dominant into homozygous recessive. Offspring, all heterozygous hybrids, offspring called F1 generation, genotype and phenotypic ratio is all alike. F1 monohybrid cross. So how the cross happens is what is it showing? It is that if a heterozygous cross is done, so what will be the case? So we will get the genotypic ratio 1 is to 2 is to 1 and the phenotypic ratio 3 is to 1. That is, when we cross two round says which is heterozygous of nature, we will get the genotypic ratio 1 is to 2 is to 1 and the phenotypic ratio 3 is to 1. F1 monohybrid cross, let's review it. Heterozygous into heterozygous, we will get 25% homozygous dominant, 50% heterozygous, which is also dominant. Okay, 25% homozygous recessive. Offspring called F2 generation. Genotype ratio is 1 is to 2 is to 1. Phenotypic ratio is 3 is to 1. What do the peas look like? Some of these peas may have a smooth texture while others are wrinkled. And now the test cross. Mendel then crossed a pure and a hybrid from his F2 generation. This is known as F2 or test cross. There are two possible test crosses: homozygous dominant into hybrid, homozygous recessive into hybrid. Okay, always remember, when we cross a heterozygous plant with a pure line or a homozygous plant is called a test cross. F2 monohybrid cross first. Trait: seed shape, alleles, round, wrinkled, cross round seeds into round seeds. And here will be round seeds and wrinkled seeds. Okay, so here it is wrong. Oh no, sorry. So it has been shown the test cross. So first he has taken the uh, homozygous dominant character and he crossed with heterozygous dominant character. So it is what I was explaining, that it is called the test cross. When we cross a heterozygous plant with a homozygous plant, it is called the test cross, and the ratio will be 1 is to 1 and phenotypic will be all alike, that is all will be round. Okay, in the second case, where we cross a heterozygous dominant character with a homozygous recessive character, we will get a genotypic ratio of 1 is to 1 and a phenotypic ratio of 1 is to 1. Okay, there will be little difference because when we are crossing a heterozygous plant and a homozygous recessive plant, we will get both the round and wrinkle shaped seeds. F2 monohybrid cross review. Homozygous into heterozygous hybrid. Offspring 50% homozygous, capital R or small r, 50% heterozygous, capital R, small r. Phenotypic ratio is 1 is to 1, called test cross because the offspring have same genotype as parents. So practice your process, work the P1, F1 and both F2 process for each of the other 7P plant traits and give me the answers in the comment section. So Mendel's law, we will continue it in the next lecture.